Welcome to NPTEL online certification course on Pharmacognosy and Metabolic Engineering. This is lecture 61, where under the broad domain of Phenolics, I am going to discuss the metabolic engineering strategies applied to shikonin pathway, aiming to produce more shikonin in the plant Lithosphaermum erythrorhizon. So, that is our main, and with once I finish this class, with this basically I will end the Phenolics. So, now go to the concept covered. So, metabolic pathway of shikonin production, although I have discussed this in the previous class. Then we will talk about the manipulation of shikonin pathway by expression of heterologous genes, and finally, we will also try to see our current understanding on the shikonin pathway. As I have shown you in the previous slide, that shikonin is basically a naphthoquinone pigments, where the one is coming from the phenylpropanoids shikimate, and the another one is the phenyl. So, this is naphthoquinone pigment. Which is red in color, accumulated in the roots of Lithosphaermum erythrorhizon, and I have also shown this one, that is the shikonin production by Mitsui Petrochem Private Limited. So, that is the first plant cell culture based product produced by the industry. Now, what I have ended the last class is basically the one root is coming from this phenylalanine root, that is the phenylpropanoid, which is basically producing para-hydroxybenzoic acid, and the other root is basically coming from the evalonate pathway. So, which is from the acetyl CoA, and which is producing the geranyl pyrophosphate or GPP. So, both GPP and para-hydroxybenzoic acid joins, and eventually the pathway moves towards the production of shikonin. Okay. Now, this have also I have mentioned that shikonin accumulated in the root. So, that is why it is red in color, and then under the dark condition shikonin accumulation happens, and also the hairy root cultures are capable in producing shikonin or using particular medium, say for example, M9 medium is very good for induction of shikonin here, and you see the shikonin accumulates in the epidermal region. So, that is why the red coloration here, but here there is no color. So, that means that when shikonin started accumulating, it accumulate in the epidermal cells. Okay. Now, we will talk about the genetic engineering of shikonin. So, now one interesting work is about discovery of a gene which is called UBIC gene. So, UBIC gene basically encodes an enzyme which is called chorismate pyruvate lyase. Or we can call CPL. This is basically from the bacterial source. So, chorismate pyruvate lyase, what it does? It produces chorismic acid, from it produces 4-hydroxybenzoic acid from chorismic acid. So, chorismic acid was used as a substrate, and which produces para-hydroxybenzoic acid as the product. 4-hydroxybenzoic acid as the product. So, the enzyme is basically the CPL CPL or chorismate pyruvate lyase, and in this process a pyruvic acid is released, and this CPL is basically encoded by the gene which is called UBIC gene. So, UBIC gene encodes chorismate pyruvate lyase. So, chorismate pyruvate lyase converts chorismate to 4-hydroxybenzoic acid in a single step, and this gene, once it is available, so they have tested this gene in tobacco. So, in the transgenic tobacco, expressing CPL enzyme, I put enzyme here, it produces huge amount of 4-hydroxybenzoate. So, now what I have shown in the previous slide, that 4-hydroxybenzoic acid acts as an intermediate for this pathway. So, for this cyclimate pathway. So, what I have shown that 4-hydroxybenzoic acid that basically joins with GPP and produces geranyl hydroxybenzoate. Geranyl hydroxy benzoic acid, or we can call GBA. So, this GBA subsequently makes the shikonin. Now, 4-hydroxybenzoic acid is basically produced from cinnamic acid, or transcinnamic acid, and that basically coming from phenylalanine, and this phenylalanine is coming from prephenic acid, biogenate, and prephenic acid is basically coming from chorismic acids. And chorismic acid is coming from cyclimic acid, right. So, this is the pathway. Now, putting the CPL here will convert chorismic acid to 4-hydroxybenzoic acid in one step. CPL, okay, and the endogenous enzymes are also there in this pathway. For example, here the PAL is there, okay, other things are there. So, if the UBIC gene which encodes CPL can be transferred into Lithophthirmum erythrorhizon, then what will happen? The existing pathway from chorismate to 4-hydroxybenzoic involved several intermediates. Now, that can be subsequently shortened by putting this step here. Okay. Then what will happen, that maybe the normal route will operate, but if you express the UBIC under the control of a strong promoter, then what will happen, it will drive most of the chorismic acid towards the 4-hydroxybenzoic acid, and you will find more 4-hydroxybenzoic acid is produced. If more 4-hydroxybenzoic acid is produced, then what is going to happen, that will get the substrate, so the pathway will move more efficiently towards the formation of shikonin, and eventually if with a bit of luck you may be able to get more of shikonin here. Okay. So, that is basically the rationale. So, then what they have done is basically this Lutz Heitz group from the University of Tübingen. So, they basically made the construct. So, that means they put the UBIC gene under the control of a promoter and a terminator. So, they have used a strong promoter which is P top promoter, P top, which is a modified 35S promoter. The promoter P top is added here, which is called P top, which is modified CMV, okay, modified S promoter, and they put a transit peptide sequence, so that it will drive the product inside the organelle. So, and the terminator should be there, and this is the transit peptide, this is the promoter, and this is the coding gene. And why they put the transit peptide? Because the product to be transformed, the protein to be transformed inside the organelle, particularly endoplasmic reticulum, so where the reaction will occur. Now, so they have used normal CMV promoter as well as the much stronger version, and what is going to happen as a result of that. So, we are going to see. Okay. Another thing is this, that okay. So, this is what is the pathway which I have shown in the previous one. So, the chorismic acid in one step makes 4-hydroxybenzoic acid, this one, this is what is this route. So, this is 4-HBA, but the normal pathway is basically through this route. Now, what they have done? So, they found that significant increase of 4-hydroxybenzoic acid upon expression of CPL. That means, when CPL is expressed or UBIC is expressed and it encodes the CPL enzyme. So, CPL successfully converts chorismate to 4-hydroxybenzoate. And then that may leads to more production of 4-hydroxy geranyl benzoic acid, but the end product formation was not marginally affected. So, it did not make much sense on this shikonin, slight increase of shikonin. So, what they also try to understand here, that what is the contribution of the existing phenylpropanate pathway. So, in order to check that, basically they did a basically inhibitor feeding, so which is AIP. So, AIP competitively block the phenylalanine ammonia lyase. As a result of AIP blockage, what is going to happen? If the AIP is blocked here, then this pathway is blocked. So, if CPL contributes to this pathway, then by blocking AIP you will see some formation of 4-hydroxybenzoic acid. Otherwise, if the more 4-hydroxybenzoic acid is not because of the CPL, but maybe because of certain other epigenetic modification, then it will not really make any sense. So, that is why they have done feeding of AIP. So, upon feeding of AIP, what is going to happen? If it blocks here, then it blocks here, then there will be some production of shikonin if the pathway moves in this direction, and if the pathway is not moving in direction, then the total shikonin pathway will be blocked. So, here they have nicely put the outcome of such experiment. So, you see here that this is control root line where no UBIC gene is here, and this is basically the transgenic root line where UBIC gene is there. So, one is the plus AIP, that means here in the control root line they did feeding with AIP, and the minus is that no feeding. So, when they have done feeding with AIP in the control root line, what is going to happen? If you see the control pathway, that means if I draw this one, so think about that this is non-existent. Think about this is not non-existent. So, here in the control, here you have done AIP feeding. So, what will happen eventually? The shikonin content will be less. So, the 4-hydroxybenzoic acid will be less, okay, in the wild type. So, what is happening here? You see that when AIP they fed 4-hydroxybenzoic acid glucoside content is very less, shikonin content is very less as compared to no AIP feeding control, where you see some amount of shikonin formation along with 4-hydroxybenzoic acid. Okay. And now, what happened to the transgenic line where UBIC is expressed? So, there what happens, normally without AIP you will find formation of shikonin in pretty good amount, and 4-hydroxybenzoic acid glucoside is also formed. But when the AIP was fed in the transgenic line, what will happen? The shikonin content is now less. If you compare with this one and this one, this is less. Not only that, the 4-hydroxy benzoic acid glucoside content is also less. That means what? That means both the pathways are contributing to shikonin formation. Both the pathways contribute to shikonin formation. That means, this pathway contributing to shikonin formation because of the transgenic, and this pathway is also contributing to shikonin formation as the wild type. So, in the transgenic line both the things are active. So, even if you block AIP, the shikonin content is reduced because this route is not coming, only this route is active, and that is why you see the content is getting suppressed both in this one. And this is what the hairy root line without AIP, it is producing red color shikonin accumulation, but with plus AIP in the control, no coloration. Similarly, here also that the transgenic line without AIP, it is more coloration, whereas with AIP the color has been reduced. So, this paper was published in the Plant Molecular Biology by the Somar et al. So, Somar was a postdoc, and Somar probably was a PhD student. So, it is Lutz Heid was the group leader from University of Tübingen, Germany. So, they published this paper. So, I think I have explained the things in a simplified way. No further discussion I will go, rather we will go to the another aspect, the Lutz Heid group. Then next, once they have checked the UBIC, now they have utilized another enzyme which is UBIA. Now, what is UBIA? We need to see. UBIA gene basically encodes a plant 4-hydroxybenzoate geranyl transfer. Okay. No. In plants, in plants 4-hydroxybenzoate HBA or 4-HB we write 4-HB geranyl transferase. So, 4-HB geranyl transferase, what it does basically? It basically joins 4-HB and GPP. So, it joins GPP and 4-HBA and it produces geranyl benzoic acid. Okay. So, this gene is called 4-HB geranyl transferase, or we can say 4-HB geranyl transferase. Okay, now 4-HB geranyl transferase, the properties of this is that this is basically a membrane bound enzyme and this is located in the ER. Endoplasmic reticulum, and it is basically regulated by light hormones, elicitors and media composition. Now, enzyme activity correlates with shikonin formation, that is more enzyme activity, more shikonin, and this enzyme was partially purified however, no structural gene was cloned. And the last point is this, that this is highly specific to GPP, but not FPP. So, therefore, the 4-hydroxygeranyl transferase is there in the pathway, but gene is not available in the hand, but its property is known. Therefore, the scientists they decided that to hire a similar enzyme from E. coli, that enzyme is basically from the E. coli. So, a bacterial hiring a bacterial UBIA gene which encodes 4-hydroxybenzoate polyprenyl transferase, which encodes 4-hydroxybenzoate polyprenyl transferase. And although this is also a membrane bound enzyme, and it has broad specificity, it accepts both GPP and pp. Therefore, they decided that to use the UBIA gene, because there is no plant gene available and it is more or less similar, but it is a more broader substrate specificity and it is also membrane bound. So, to transfer this gene into lactospermum may do the work. So, the UBIA gene was taken from the E. coli, the UBIA, which is 4-hydroxybenzoate generanyl transferase, which is basically this is the plant gene. 4-HB generanyl transferase is the plant gene and this is basically the the bacterial gene, and they have expressed again in the hairy roots using co-transform hairy roots, and the construct is very interesting. So, that is why I would like to tell a little bit about this, about the gene construct. So, because they want to put it inside the endoplasmic reticulum and to stay there. So, they have used a fusion of UBIA with H del. So, this is basically a fusion of UBIA H del. So, how we write UBIA H del, and then they put a signal peptide and they put the promoter. So, this is a very strong promoter we use, which is called O3M promoter, which is octopene synthase 3 with a manopene synthase promoter. That is a very strong promoter, and then they put a terminator here, NOS. Now, the purpose of putting is that when they put the H del, so H del is basically is basically a tetrapeptide. They have taken and that actually function as signal retention inside the endoplasmic reticulum. So, it function as a signal for retention of soluble and membrane bound protein inside the endoplasmic reticulum. So, this leads to the retention of soluble and membrane bound protein inside the ER. Okay, one small correction I would like to say, in the previous one, when I talked about this one. So, the construct which is the transit peptide. So, this transit peptide was used basically to transfer the protein UBIC, that is the CPL protein, to move inside the not endoplasmic return, but mostly inside the plastid. So, please make this correction. Okay. So, this will return inside the endoplasmic reticulum. So, because the pathway, late pathway are in the endoplasmic reticulum. So, that means when this gene is active, so then what is going to happen, that this UBIA will make this GBA, which is 3-geranyl 4-hydroxybenzoic acid. This subsequently converted into geranyl hydroxygeranyl hydroquinone, and then one pathway moves towards the formation of shikonin and the other pathway moves towards the formation of ecneophiran. And so, the outcome is this, that there is only marginal increase of, although the gene has worked well, and as a result of that, this product 4-hydroxygeranyl 3-geranyl 4-hydroxybenzoic acid, this product produced in huge amount. The enzyme activity was very consistent, but eventually the shikonin formation was not that much affected. So, what it indicates that perhaps you need to know more about the more downstream pathway. So, the downstream pathway, that is the downstream pathway is important. Exerts more control. Okay. Now, the third one is by the same group what they have done, another paper they published in 2002. This time they have taken HMG-CoA reductase, which converts HMG-CoA to mevalonic acid, because that is the route which contributes to the GPP formation, and they have taken the UBIC. So, UBIC converts corismate to 4-hydroxybenzoate, so that you get more of 4-HBA, and mevalonate is basically HMG-CoA reductase. If it work, it converts HMG-CoA to mevalonate. So, more of GPP. So, it is expected that more GPP will be produced, more 5-hydroxybenzoic acid will be produced, and as a result of that the pathway will move in this direction, and eventually it produces more of 3-geranyl 4-hydroxybenzoic acid and eventually more of shikonin. So, again the shikonin increase was marginal, although the 3-geranyl 4-hydroxybenzoic acid is produced in substantial amount, but the final product formation was not that much. But the metabolic engineering attempts on the design strategies what they have adopted, that is means scientifically that was very well designed, but however, scientists could not see the expected result, because more complex regulation exists in the downstream process. Now, in addition to this, what scientists they are also attempting, they try to see if any transcription factors like EIL1. So, EIL1 is a transcription factor. So, that perhaps plays an important role in the regulation of the pathway. So, they made an attempt to express EIL1 as different ways. CK as one is the control, this is the control wild type, and EV is basically the expression vector, that is only the expression vector to check it worked or not. Then the third one is basically the EI, where basically EIL1 they try to block it. So, it is basically the EI construct. You see that there are RNAi approach. Okay. And the fourth one is basically the overexpression construct. So, they put overexpression construct, this is EO. Okay. So, EV is basically the vector, simply the expression vector, the EI is basically the blocking of the EIL, and the fourth one is basically the overexpression. So, what you see from the root coloration itself, that this leads to more coloration, the right one. So, that indicates that overexpression strategies worked well, and what we see here also, that when you put it in specific medium, so first one is the normal growth medium and second one is the say M9 medium. So, what you see here, that the content of this product is much higher in the overexpression construct, where you see more of shikonin formation there, then and EI of course, it is very less, EV is empty vector. So, it is unaffected, just the it contains a gas gene or GFP gene. So, that function as a wild type. Okay. So, what scientists they predicted, that this strategy worked well, and like overexpressing a transcription factor like EIL1 indeed makes sense in the production of shikonin in Lithosphaermum erythrorhizon hairy root. So, this is the PGT. So, this says that basically there is an upliftment of this enzyme, this step, this is what is that UBIA, what we call when you use the bacterial gene, but the plant gene was this geranyl, this 4-hydroxybenzoate geranyl transferase, which makes geranyl para-hydroxybenzoic acid, and eventually it makes the shikonin. So, what they checked the expression of these enzymes, and they found that expression got uplifted. So, basically this is another update of this shikonin biosynthetic pathway. What you see that there is a cell wall, and then outside the cell wall there is the apoplastic space, and then shikonins are basically coming out, and when it is cell culture it secretes in the medium. So, it is basically coming out. So, it is not known whether any transporter involved in that, and scientists made different attempts, like they try to block here, they try to block here, and they see that how the regulation happens. So, what we have learned is this, that the pathway is this 4-hydroxybenzoic acid and GPP, which makes this geranyl hydroxybenzoic acid, then it makes geranyl hydroquinone, and then from there the cytochrome P450 will make 3-hydroxygeranyl hydroquinone, and from there maybe the pathway produces intermediates leading to the deoxyshikonin, and then ultimately the shikonin, whether the other pathway moves towards the dehydroshikonofuran, and which makes the ecnofuran. So, this is the in general the pathway, and so our current understanding is this, that we know this much, that this one we know this pathway contributes, we know this pathway contributes, we know geranyl hydroxybenzoic acid, then geranyl hydroquinone, then hydroxylated geranyl hydroquinone, and in the latest information is this, that this cytochrome P450, which converts geranyl hydroquinone to hydroxylated geranyl hydroquinone, has been characterized, and but what converts hydroxygeranyl hydroquinone towards the shikonin and towards the benzoquinone, this remains elusive yet. Okay. So, this is basically the hydroxylase, this is cytochrome P450 hydroxylase, which converts this hydroxygeranyl hydroquinone into the other intermediates like 2-oxogeranyl hydroquinone, and then one of these pathway contributes to the formation of deoxyshikonofuran, and other moves towards the formation of shikonin. So, we will not go into that detail, simply what you should know that the hydroxylase enzyme has been characterized, but beyond that which is not known. And much later another paper also published, where they say that this is shikonin acetyl transferase. Shikonin acetyl transferase has been characterized from the pathway. LE means lithoplasma erythrorhizome, which basically converts shikonin to other shikonin derivatives. So, it is not only the shikonin, but other shikonin derivatives that also plays important role, and in order to produce more of shikonin, it may be required in the future to block this activity, possibly in the future by RNAi or anything, still do not know, and if that is possible, then it may be possible to make more of shikonin accumulation in future. So, with this I end now the class. So, this is in a nutshell, that one cytochrome P450 has been characterized, and there may be deoxyshikonin to shikonin. Here is another cytochrome P450, which is only partially characterized, and so what you need to know is basically details of here. So, then only it is possible to make more shikonin as per we desired in Lithosphaermum erythrorhizome, and years to come when we will be able to achieve this. So, this is about in brief about the metabolic engineering strategies so far being explored for the production of shikonin in Lithosphaermum erythrorhizome using hairy root culture as a model, because it is the organ culture. So, it will be more stable than the cell culture. So, with this I end this class. Thank you.